said that we were going to cover two concepts, and one was the image formation, okay, the physics of how we bend light waves and create the image. So that's what we just did. And now we're going to talk about the physiology of how the photoreceptors are affected by the energy of light and the pathway that the nerves follow. So here's a little bit higher view, mag view of the pigmented epithelium and the cones and the rods associated with them. So again, the pigmented epithelium is not involved directly in image processing, but keeps the rods and the cones functional by recycling that photopigment. So there are two components that are used when we talk about the physiology. One is retinal, and that is derived from vitamin A. And the other is a light-sensitive pigment known as opsin. All right, so with cones, rather with rods, which are sensitive to low levels of light energy, but are not responsive, they don't respond to various color wavelengths. These are combined together to form a molecule known as rhodopsin. Okay. Now we also have opsin in our color molecule, in the cones, and they're responsive to different wavelengths of light. So the wavelengths are, the color opsins are blue and green and red. Those are the primary light colors. Those are not the same as primary colors when you mix, which are red, yellow, and blue. Okay, these are red, green, and blue. And so when we see color, it's usually a combination. In the light are different wavelengths, and so different numbers of cones and different numbers of each of these sensitive cones are activated and respond to the light, and therefore we see various shades. So the S cone, M cone, and L cone refer to the opposites. So we have short wavelength, or S opsin, middle wavelength, or M opsin, and long wavelength, or L opsin. And so these are going to be for the cones. So blue, green, and red. When we talk about the actual physiology of opsin, however, we're going to focus on the rods. So I'll come back to the color blindness in a moment. So this diagram is a little bit small, but I'm going to leave it up on the board while I talk about the activity. This is another G protein pathway, all right? So I'm going to describe what is going on when the eyes are in the dark. So you wake up at 2 o'clock in the morning and the curtains are closed and everything's dark, or you're actually asleep. And so we're going to be looking at the rods. And both of the rods and the cones have these discs. Some of the discs are within an enclosed fold of plasma membrane, or they are kind of enclosed in this type of a structure. That doesn't affect the way they function, but it's kind of just a slight difference in their organization. So here's our rod. Actually, I don't need to draw that quite that long. It is. I'm just losing my functional portion of the cell. And inside are a series of these discs. Okay. So I'm going to take one of these discs and talk about it over here. Okay. As the discs are utilized, they move upward towards the pigmented epithelia and then are endocytosed by the RPE and recycled. So they kind of sit up there with lysosomes and are just fed. It's like sitting there with their mouths open and just gobbling up these discs. So the first diagram, the first pathway I'm going to show you is occurring without light stimulus. So this is what's going on when the light waves have not reached. There is no photon energy activating the membrane. And this is probably a little hard to see from the back, but I think you have these diagrams in your lecture notes. Okay, so we're looking at, are you dead on this one? We're looking at the structure of the plasma membrane of one of the discs that is now going to be responsive to light. So here's a disc membrane. And embedded in that membrane is our rhodopsin. And it's going to have the retinal in there and the opsin. And the opsin can change shape. That's kind of what's illustrated here. All right? And so it goes from oscillates back and forth between the transform, the straight form of the retinal, or the retinal, or the cis form of the retinal. And that's just a change in the shape of the molecule that affects the G protein associated with it. So we have a G protein. And unlike our other G proteins, this one has been given its own specific name, which is transducin. In addition, there is a membrane-bound enzyme known as phosphodiesterase. Phosphodiesterase has been on some of the diagrams that I put up on the board when we were describing G protein and cyclic AMP. And its function in the previous diagrams, I didn't emphasize it a lot, was to break down cyclic AMP. Okay, so it removes in a cell that is has a stimulatory response, more cyclic AMP is produced. Obviously, we need to get rid of that cyclic AMP, or you're going to be in a constant state of excitement, just like we have to get rid of neurotransmitters so that we can respond to new neurotransmitters. So if the cell is making cyclic AMP, we have to get rid of cyclic AMP. And that's what phosphodiesterase traditionally does, is it gets rid of cyclic AMP. It has a slightly different function here that I'll talk about in just a moment. All right, so that's what we see in these discs. Just multiple transducins and phosphodiesterase and rhodopsin molecules. So out here will be the plasma membrane, the one I'm drawing kind of out that portion, and it has ion channels. It has leak potassium channels and gated sodium channels are the ones that we're most concerned with here. So in the dark, and this is unusual, the gated sodium channels are open. The reason being is because of other enzymes, the cell has a lot of cyclic GMP. GMP is made from guanine triphosphate, like cyclic AMP is made from adenine triphosphate, so it's a slightly different form of energy storage. And so in the dark, the cell has a lot of cyclic AMP. The cyclic AMP binds to the sodium channels. And these are kind of ligand-gated except on the inside. And so with cyclic GMP bound to them, they open. All right? Normally this would happen if a neurotransmitter binds to its receptor as a G protein. The G protein releases the alpha subunit, and adenylate cyclase is activated, and it changes ATP to cyclic AMP, and cyclic AMP will open other channels. Typically, they're calcium channels, but they can occasionally be other channels. So in this case, cyclic GMP maintains sodium channels that are open. Which way will sodium ions move? Into the cell or out of the cell? Into the cell. So positive sodium ions move into the cell, and that depolarizes the cell. It doesn't bring it to over positive. It brings it from a minus 70 that we were talking about to about a minus 40. Okay? We still have our potassium leak channels, but they are overrun by the positive ions flooding in through the gated sodium channels. So with all of these positive ions flowing into the cell, we create a graded potential. It's not an action potential, okay, because it doesn't reach over zero, and it's not opening our voltage-gated channels along the cell. But it's a graded potential. And so these positive ions flow through the cell. Potassium is going to be leaving, but not enough to lower the membrane potential. And they reach the dendritic end, if you will, of the photoreceptor. So I'm going to draw that a little bit larger. And when this graded potential reaches this end of the cell, it opens voltage-gated calcium channels.
Without an action potential.
Right, it's a graded potential. So it's threshold for the. So calcium enters the cell. What happens in the synaptic gluton when calcium enters the cell? The synaptic vesicles are capable of fusing to the membrane because of the synaptotagmin. And so the same thing happens here. Synaptic vesicles will be released from the photoreceptor, and their neurotransmitter is glutamate. All of this is happening in the dark. And so this graded potential that's flowing across the cell membrane and down the cell is actually identified as the dark current. Sounds like a movie that should come out. Okay. Which is kind of backwards of what you would think would happen. We're so used to have a stimulus having an excitatory effect. And here it's the lack of the stimulus that is excitatory, not to create an action potential along the cell, but certainly at the end down here, it's opening those voltage-gated channels and allowing calcium in. Yes?
The dark current is the flow through the rod.
Mm-hmm. Well, this one as well. So it starts here and then flows through. Okay? So this is happening again, in the dark. And a similar event would happen for our cones. It just takes more energy, and the opsin is a different molecule that's susceptible to the light energy. So, essentially what happens with a light photon, all of this stops. And here's how it stops, okay? So I'm going to change events here. Does anybody need pictures or diagrams or whatever?